Hey, this is 6.05, and we're going to talk about inheritance where we have multiple alleles. So what does it mean to have multiple alleles? Does that mean you get more than two alleles for a trait? Well, yes and no. There's two different ways we can talk about multiple alleles. So one is something called polygenic. And these are traits that are influenced by more than one gene. So we've got like height and skin color. You have 2, 3, 4, 5, but you have more than one gene that affects it. So you can have two or more genes that play in. Other things like some diseases. But polygenic and multiple alleles aren't the same thing. So the other thing we can have when we talk about multiple alleles is you have one gene and you still get two alleles, one on each chromosome, but there's more than two choices. So there's 2 plus choices for alleles. And the classic example of this is blood type. So the alleles that we have for blood type, start off with blood type, are A, B, and O. Now, A and B are codominant, and so we write them as a capital I with an exponent, superscript A, and we write them as a capital I, superscript B. Why? Because these are codominant. And what we can have then is the third choice is type O. And this is recessive to both A and B. So these are my choice of alleles. I have a choice of 3 alleles. I could have a type A allele, I could have a type B allele, or I could have a type O, or I could have a combination of, so I'm going to have a combination of 2 of these. Well, what is this A and this B? Your red blood cells, this is a red blood cell, and in this case, this red blood cell has a smooth surface. And we've talked about in past chapters about how you can have protein spikes that come out of the surface of a membrane. And this is really important when viruses attack cells, and you've talked a lot about protein spikes with the COVID virus. Well, it turns out you can have 2 different shapes of protein spikes. It's a little difficult to see. These are they show this as a circular. How accurate that is, I don't know. And they show these as triangles. So type A protein spikes have one shape and type B have another. So if you have both, I have triangles and circles here, you're type AB. So our phenotypes can be type A, type B, type AB, or type O. So you have 4 possible blood phenotypes. So what are our genotypes for that? Well, we know that you can be homozygous dominant. So this is going to be IA, IA. You can have IB, IB. This is going to have an A allele and a B allele. So this is going to be IA, IB. And this is the only way that you can have AB. So what about O? You have to be homozygous recessive to have O. But there's 2 more genotypes. If these are dominant to this, couldn't I also have heterozygous with an O allele that gets masked because it's recessive? Yeah, I could. Here I could have heterozygous for type B. So we have 4 phenotypes and we have 6 genotypes. And you should be really familiar with those because we're going to do a lot of blood type problems. Okay, so let's start off. We've got a woman with type A. She's got an A allele and an O allele, is married to a B person. They're heterozygous for B. what blood types will their children have? Well, first of all, let's write out this in our blood type form. So if you're heterozygous for type A, you're going to have I, A, I. And if you're heterozygous for type B, you're going to have I, B, I. So I'm going to put these alleles at the top of my Punnett square. Okay, do you want to go ahead and finish the square and see what you predict? All right, did you give it a try? Should have said, did you pause the video and give it a try? So when I fill in my letters, so these are my resulting probabilities of blood types for the offspring. And what we see is we're going to have all four blood types. I'm going to have type A, I'm going to have type B, I'm going to have type AB, and I'm going to have type O. By the way, these little hash marks, in case you haven't seen them before, means the same thing as the line above. And my ratio is going to be 25% for each of them. And my genotypes are, they're heterozygous here. So I'm going to have IAI and IBI. All right, let's try another one. A person with type B blood has children with a type AB person. What blood types are possible among their children? Okay, if you want to, pause the video here and see how you do with this sample problem. Okay, did you give it a shot? So if I'm heterozygous for type B, I'm going to have I, capital B, I. This is going to be IA, IB. I'm going to put these alleles along the top of my Punnett square. Remember, these are the possible gametes. When we do Punnett squares, we're talking about gametes. When I fill in my squares, it's going to look like this. So what is my result? Well, 50% of my offspring are going to be type B. And I'm going to have, of these, 25%. So it's 25% of the total. If you take this 50%, you'll have half and half, are going to be homozygous dominant. And half of them of the whole is going to be heterozygous. I'm going to have 25% type A. And that is heterozygous. And I'm going to have 25% AB. So type O is the most common. And I believe it is AB, I don't remember if it's positive or negative, is the least common. But think about, before we move on to our last sample problem, let's talk a little bit about what it means for these spikes. So we talked back in chapter 2 about if we have carbohydrate chains or protein spikes, that those serve as name tags and that the body recognizes protein spikes that belong to your body. And if you get something in your body that your body doesn't recognize, it will attack, it will destroy those cells and you'll get an inflammation reaction. So if you're type O blood, your body says that your blood cells should all be smooth. So if you go to the hospital and they give you the wrong blood type, so if they give you type B blood and they give you a blood that has spikes, your body sees those as an invader and it's going to attack those and kill them. And when it happens in your blood, it's really serious. You can die if you get the wrong blood type. That's why if you go in for a surgery, they will take your blood type twice. 2 different people will check your blood type and they'll put it in 3 or 4 locations to make sure that you do not get the wrong blood because that can be fatal. So hospitals really guard against that. If you have type AB, you are not worried if there's no spikes. You're not worried if you have B spikes. You're not worried if you have A spikes. So here's the takeaway from this. If you have type AB blood, you can receive any blood. Get a different color pen. So this can receive any blood. So it's called the universal recipient. If you have type O, you don't have a protein spike that's going to affect anybody else. And so this is known as the universal donor. Now, it's a little more complicated than that. If you've gotten your blood type, you'll see that there's A positive or A negative, B positive or B negative, O positive or O negative. And that it's called a rhesus factor. So that is a whole nother set of proteins. And so you can have O negative or O positive. If you're O negative, you don't have any spikes at all. If you're O positive, you have a different set of spikes. So actually it's O negative that's the universal donor, and it's A positive that's the universal recipient. But we're not going to focus on that so much. So let's go ahead and let's do one more practice problem. So I'm going to go ahead and slide my camera down. Go ahead and pause the video and see how you do with this. Okay, a person with type O blood, so they're going to be homozygous recessive, is married to a person with type A blood, unknown genotype. So they're either type A, or AI. They have 6 children. 3 of them have type A. So we have 50%. So our offspring over here, we have 50% type A. And 3 of them have type O, 50% type O. What is the genotype of the 2 parents? Did you say this? If the parent that has type A is homozygous dominant, can I ever get any type O's? Nope. Let's do a Punnett square to prove it. Fill in our dots or boxes. And sure enough, there's our 50% type O, our 50% type A. All right, there are going to be other traits we're going to look at, like coat color in chinchillas, and like some of your practice problems, it might be the snake, patterns in snakes. So there's other examples of multiple alleles. So what are our takeaways? Our takeaways here are one of the most common ways of talking about multiple alleles is blood type. Don't confuse it with polygenic. Polygenic means you have multiple genes that can affect these traits. For multiple alleles, you still have one gene, you get 2 alleles, but you've got multiple choices. So for example, when we do the chinchilla problems, there's 4 possible alleles you can have that affect the chinchilla coat color. And we talked about the fact that these correlate to protein spikes when you're talking about blood type. You have A and B that are codominant, you have O that's recessive. So you're going to get a combination of these 6 genotypes, 4 phenotypes, and make sure you use the right notation when you write them out. Right, so you've got some practice problems to do with these. Good luck, and I'll see you in the next video.